Hi, my name is Pourya Naderi, and I'm happy to present our work on the analysis of multi-pathway targeting by microRNAs to the ISMB community. MicroRNAs are small non-coding RNAs that act as post-transcriptional regulators of gene expression. They often target dozens of genes across multiple pathways and biological functions. Understanding the regulatory roles of microRNAs across groups of biological functions is critical to elucidating their role in cellular processes and disease programs. But current methodologies to infer microRNA pathway interactions only focus on individual and one-to-one microRNA pathway relationships. So we asked, how can we identify multi-pathway transcriptional programs under the regulations of microRNAs? We developed a systems biology framework called PanomiR to identify pathways that are associated with disease, that are dynamic and show differential activity across disease conditions, and find the microRNAs that target groups of these pathways to interact. Our system maps gene expression data combined with known pathways into pathway activity matrices. By using linear models, our system identifies pathways with differential activity between disease states. Using a previously established tool, Pathway Co-Expression Network, we find modules of coordinated pathways that correlate in disease conditions, and we place pathways with differential activity in certain groups. Then we have developed a statistical test to define microRNAs that target coordinated groups of pathways. To evaluate the utility of our system, we applied PanomiR to two distinct case studies. The first one was from the liver cancer gene expression dataset from the TCGA. PanomiR found 3 clusters of dysregulated pathways in the TCGA liver cancer data. These included a group of pathways related to upregulation of transcription, a group of pathways related to upregulation of cellular replication, and a group of pathways related to dysregulation of signaling pathways. It assigned distinct microRNAs to each group of pathways. For example, it assigned miR-107 and 124 to the cellular replication cluster, which have been previously suggested to regulate cellular replication in liver cancer. One common bias of standard methodologies when studying microRNA pathway relationship is a known bias of prioritizing microRNAs with a higher number of targets. So we compared PanomiR with standard methodologies, including enrichment analysis and p-value aggregation methods for multi-pathway analysis, including Stauffer's method and Fisher's method. We showed that, as expected, enrichment analysis, Fisher's method for p-value aggregation, and Stauffer's method have a bias towards prioritizing microRNAs with higher number of targets, whereas PanomiR, as it uses an empirical test, did not have any correlation between the number of targets by a microRNA and the rank of microRNA for targeting pathways. In the second case study, we applied PanomiR to an Alzheimer's disease dataset from the temporal cortex of Alzheimer's disease patients from the Mayo Clinic cohort. In this dataset, PanomiR found 2 major clusters of pathway dysregulation. One was downregulation of RNA metabolism, RNA degradation, DNA repair pathways in Alzheimer's disease, and the second one was related to upregulation of several signaling pathways, including TGF-beta, GSK3, NF-kappa beta, and YAP1. We found that microRNAs that have been prioritized by PanomiR to target the group of upregulated pathways have been strongly suggested to have therapeutic potential in Alzheimer's disease. For example, miR-155 is now in clinical trials for targeting TGF-beta pathway in Alzheimer's disease. Other microRNAs of interest include miR-27, miR-LED7B, miR-145. PanomiR is available as an open source R bioconductor package, along with an open access manuscript published in Briefings in Bioinformatics, which you can access using the QR codes provided here. Finally, I would like to thank our collaborators and our funders for making this study possible. Please feel free to contact me via email or via Twitter or X using the handles provided here. Thank you very much.